Well, welcome everybody to the distinguished lecture series. My name is Jeff McDonald, and I'm the Associate Director of the Global Institute for Water Security. And it's a real pleasure to welcome you back and to introduce our speaker this week, Professor Andrea Rinaldo. Before we get started, I just want to acknowledge that we're coming to you from Treaty 6 territory, homeland of the Métis, and we reaffirm our relationship with this First Nations group and their ancestors as we work to deal with problems of water sustainability in a cooperative way. This is underwritten by the Global Institute for Water Security and also the Global Water Futures Project, and we thank both of them for their sponsorship. I also want to acknowledge that this serves as the so-called 990 credit for the Masters of Water Security students, some of whom are in Beijing and watching this in a recorded setting, and welcome to you as well. But anyway, today I couldn't be any happier to introduce our speaker, a real hero of mine in the field, Professor Andrea Rinaldo. He's Professor of Hydrology and Water Resources at the École Polytechnique Fédérale Lausanne, usually known as EPFL in Switzerland. This is really a top institution in Europe, and he's the director of the Laboratory of Eco-hydrology. Andrea holds a number of concurrent posts at the University of Trento in Italy, University of Padua, and I won't go into those details, but I would like to say that Andrea really is the international thought leader in, I'd say, transport phenomena in the hydrologic cycle. You'll hear today about ideas on networks as it relates to pathogens and disease, but Andrea's had deep impact on our field in these aspects as applied to geomorphology, stochastic modeling in hydrology, and eco-hydrology. And for that, of course, he's received probably, I think, more recognition and deep recognition, deservedly so, than anyone I know. A very brief list of some of the more pertinent awards. He's Hydrological Sciences Award from AGU. Of course, he's a fellow of AGU, Dalton medalist from the European Geophysical Union. In 2010, I think together with Ignacio, won the Prince Sultan Abdulaziz International Water Prize, the Creativity Prize, which is really the big one. And that is really quite remarkable. I think what's most remarkable for me, he's a fellow of the Royal Swedish Academy of Sciences and a foreign member of both the US National Academy of Sciences and National Academy of Engineering. I don't know of any other hydrologists with that level of recognition. His contribution to the field in terms of service is also enormous. He served as editor of Advances in Water Resources from 2011 to 2015. And since 2014, he's been editor of PNAS, a journal we all know and love. I think beyond that, and maybe most importantly he's just a delightful colleague, collaborator, and human being. I aspire not only to come somewhere possibly within his orbit scientifically, but also to be as dapper and charming as Andrea. He's just one of the most delightful people we have in our field, and I'm so glad we finally got him to give this lecture. I've been trying for many years, but he's a super busy guy. Anyway, Andrea, over to you, and we look forward to hearing from you today.
Thank you so much, Jeff, for this generous introduction. It's wildly exaggerated. I'm pretty sure I will be disappointing after such an introduction, friends, etc. So thank you so much for the invitation, which is very dear to me. As you said, I promised I would do it, but I maintain my promise to be at the global, in person, visiting you. So let me start to share my screen. Okay. So here is my title. Let me give you a brief introduction. The title is some sort of I'm trying to look at a coherent eco-hydrological perspective of global water security, seen in a somewhat different angle from traditional hydrology, which is mostly concerned with floods, droughts, but key on fair distribution of water. That's what I care for. And I see that from a narrow perspective as hopefully not as narrow as one might imagine, seen from a long experience we've developed in studying how nature works, seen through the structure of the networks and how they function as ecological corridors. And the picture, I took this picture of the banks of a Magna River, and well, not a Magna, it's one of the tributaries almost flowing into the Magna, one of the big tributaries in the majestic deltaic system of the Ganges Brahmaputra in Bangladesh. And mighty waters, as you might say. But what makes this picture significant for what I'm trying to demonstrate is that this picture is taken maybe 300 meters downstream of the largest diarrheal disease hospital in the world. In Matlab, seriously, Bangladesh, is an outpost of a famous center based in Dhaka, which is a place where cholera, epidemic and endemic cholera developed in the first place many moons ago in an evolutionary sense, and from there irradiated through several pandemics worldwide. Why is this relevant? Because this guy was telling me that, was just showing me in as much as it happened to John Snow in London, 19th century. But it couldn't be that the mighty waters of the Magna River would cause cholera. Now, given that 200 meters upstream, you have one of the, you had something like maybe 350 current diagnosed infections of cholera. You wonder whether the guy would catch it. Now, you catch cholera, and I apologize for the long introduction, but it sets a tone for what I'll be trying to show you. And in any case, don't worry, I do have a buzzer going on when time comes to go to my conclusions. But you catch cholera if you ingest enough bacteria. Enough means something which depends on your body weight, on number of conditions, so the strain of cholera bacteria that we are talking about. And if you think in retrospect, answering the trivial question, will the guy catch cholera? And you see how complicated it is. Because you have to know, essentially, whether locally in one point in space and at that precise time, the concentration of free-living bacteria shed from the leaky sewers of a hospital 300 meters away and dispersing through a river would generate a local concentration such that the guy would catch cholera. So essentially the question is, we don't know whether the guy would catch cholera. It would be way more prudent if the guy didn't drink that water, but remarkably, the weakness of our prediction, our capability to predict, is a permanent liability that we do have. And what I try to explain is essentially that working with river networks may in fact help reduce the uncertainty which is inherent in the processes, and perhaps to rethink distributive water justice. So the kind of questions, this is a picture I took again. I love taking pictures, because in the field work we did instead in Burkina Faso, it's a young woman passing on to one of the 15,000 small dams that were being constructed there in recent times. But the general idea of it is, what do I mean by global water security? What do I mean by looking forward towards a fair distribution of water? To me, it means that we should be able to answer relatively simple questions, but with plenty of background. But is that, will future large-scale water resources management plans include, say, the reduction of biodiversity loss in river networks? Will it do it? Because now they currently don't do it. Or for that matter, are river networks templates for the spread of waterborne or water-based disease? But whether we, by looking at the river networks, we would know the avenues of the propagation of the disease in a broader sense, in fact. Or are we capable, and that's another point, of making a compelling economic argument for preventing water development schemes in the light of predicted increases in the burden of, say, poverty reinforcing debilitating diseases they would bring? Or are we capable of really understanding biological invasions of foreign species of different kinds, etc.? What I'm seeing these, now pandemics are very much in the news still, but the idea is that, can we learn a lesson relevant to even airborne disease like COVID from a structural river network. So essentially, I'm trying to demonstrate, try to show you, to convince you that river networks are indeed important in the quantitative evaluation of ecosystem services. So our weakness and the liability we have is the fact that in the past, we haven't been able to quantify seriously pros and cons and consequences in the evaluation of ecosystem services. So essentially, we're talking about a little bit of science in a nutshell, without technical details, but I'd be delighted to speak about the technicalities at some other time, about directional dispersal and what it means, and spatial ecology, and how in fact form and function of river networks meaning fluvial ecosystems that are teeming with life and services and supporting as an ecological, as a substrate for ecological interactions, biological invasions, population migrations, or again, the disease. And that's more or less what I'll be trying to do. And let me, I'm sorry to be embarrassing, but it happened, actually, this is contained in a book I published recently, but practically no one has seen it because the publisher, differently from the past, didn't do any advertisement nor got any review for it. It's called, is the title of that, and you see that the front page sports the picture I just showed you. In any case, it's in there you got all the technical material. So my first point of discussion is the following. Suppose, like in this case, you clear a mangrove swamp to make way for a commercial center. A great thinker, Partha Dasgupta, in fact, analyzed the case of what do we mean by how do we account for the depreciation of natural capital, and how this affects the debate between development thinking and environmental thinking, to the point that Partha concluded that development thinking is stacked against nature. What do I mean with that? It means that if you make way by destroying a mangrove swamp to make a commercial center, in the GDP or in the economic indicators like gross domestic product or whatever, see employment, richness measured by any scale, in the following years, you will certainly see the benefit of a commercial center. But you do not see the costs that are generated with the loss of ecosystem services the mangrove swamp was providing, from defense, coastal defense, from production of fiber, from fish nursery areas, to cultural services, and in general, people were still obsessed by the so-called the curse of the Kuznets curves. Kuznets was the European expatriate in Harvard who won the Nobel Prize in economics, by somewhat supporting the curves that, in a sense, were suggesting that the richer you are, the more these inequalities are reduced. It is the degree of inequality you would have in a place would be reduced by simply the growth in indicators like a gross domestic product. Now enter Thomas Piketty, and a neighbor in nearby now in Paris, with his capital in the 21st century. And it turns out that not that Kuznets was wrong, but his data were very much biased by the fact that it was post-Second World War. And in reality, it's simply false that the richer you are, the more inequalities, and thereby also fair share of water security are reduced. And one of the things that we need to realize is that unless we could put a price tag, no matter how unclassy this might be, unless we can price the planet or put a price tag of ecosystem services, it means that something which is, like in this case, invaluable, means this is worth nothing in economic terms. And I contend that river networks can help in quantitative evaluations of this kind. And economic indicators change dramatically when the depletion and the degradation of natural resources and their ecosystem services are accounted for. Unfortunately, the natural capital tend to be undervalued because it is unique. It is well, hard to put the price tag on, poorly understood, and difficult to measure overall. So consider this example. This is a group of distinguished scientists, a couple of them very good friends of mine, of Swiss Tropical Institute in Basel. They did a meta study on the relationship between a neglected tropical disease, schistosomiasis, and water resources development are very much related. Schistosomiasis, and I'll be telling you slightly more if I make it later on, is a chronic parasitic waterborne disease that affects something like 800 million risk of contracting the disease every year. And a stunning 90% of a current schistosomiasis is localized in sub-Saharan Africa, by the way. And it's a strange disease because once you get it, you can clear it out by taking praziquantel, but it doesn't give you any acquired immunity. So anytime a kid goes back to the water, it gets infected, and it's hard to keep kids. This I took a picture in next to Ouagadougou, where we did the field work I'll be briefly talking about later. But what is interesting is that this paper collects compelling evidence gathered by study that examine the relation between the water resource development projects and the spread of schistosomiasis, primarily in that particular part of sub-Saharan Africa. And this is clearly related to the expansion of a range for the intermediate host of the ecological cycle of the thing. It's a mandatory intermediate host, which is some species of freshwater snails, which requires quiet waters to be able to settle. And I'll be showing you the diagram later on. And so essentially, what happens is that in waters infested with a certain stage of the life cycle of the parasite, you get infested by simple penetration of the skin. Now, what happens is that 15,000 small dams have been created in Burkina Faso, and the GDP and the agriculture, well, absolutely sensitive to that. You've seen that, you've seen how these generated a small economic boom for the country. But at the same time, what it doesn't show is the fact that this is a poverty reinforcing, debilitating disease. Because what happens is that kids that get that get all kinds of learning impairment and all kinds of costs that have to be quantified that are not accounted for in the development of Burkina Faso. So this is at the heart of what I believe it's water security. So you spend a hell of a lot of money, you develop huge irrigation networks, which widely expand the range of intermediate hosts, and you bring richness, good crops, etc., and disease. And why can't we quantify whether this was justified or not? Because in the past, we couldn't predict what would be the effect of a water resource development scheme. So we can't have a mitigation of mitigating effects become integral parts of the planning, of the implementation, and the operation of future water projects until we'll be able to do that. So essentially, what is sustainability in modern terms, even by the standards of modern environmental economics? Is the social worth of the entire set of capital assets that include the natural capital, and that defines the wealth and the poverty of nations. No economy can be made to thrive by mining continuous natural capital. And what I'll be trying to show you is that, in fact, ecological processes, when operating on a substrate, which is a river network, are so constrained by the shape and by the form and by their function that in reality some predictability is granted, much to our surprise, I have to say. And to try to convince that, I'll be briefly tell you what kind of, by the way, it was a kind of a trick, because this is, I mean, for those of you more, this is not a river network, this is a tidal network, and in the Lagoon of Venice, in which I keep returning to my home place, in fact. But the concept applies no matter what, something whose geometry and topology and metrics we can actually define. So you're talking about the fluvial landscape per se. We have remarkable capability to remotely acquire and objectively manipulate the description of natural landforms over several orders of magnitude, to the point that if I take out the scale bar, you cannot tell whether this is a small creek next to Saskatoon or this is the Amazon. Why? Because nature tends to produce the same shapes, and that's at the heart of a debate which was foundational to the statistical physics in the 80s, something in which essentially remotely you send a beam and you get an elevation over a certain square. Over a certain square, you get a reasonable approximation of the elevation of that, and you can generate by steepest descent, calculate, say, elevations, gradients, curvatures, in something in which essentially given by elements of something which currently undergraduates can manipulate fairly well, and that besides are featured in ArcGIS and these geographic information systems of many kinds. So essentially, you can generate a very coherent extraction. For instance, this is a network of concave sites which you have in a particular place, so places which you have like a concave up colluvium in geologic terms, or the subset of that, which generates something which brutally thinking is that you can label each of those pixels that make up your landscape you have remotely acquired with the channeled and the unchanneled parts, because they do have ecological significance, of course. And we have a number of tools which are sophisticated but interesting. For instance, you have networks for which you have exact results, which is a great advantage. So I have to do calculations. And one is the so-called Peano network, that is over a curve that fills the plane, was the title of his paper 1870 something, which was absolutely shamed by the mathematical community of his time. He was still working on it, which is an important network. It's generated by recursive procedures, so we know exactly how to characterize that. And for instance, the width function, for instance, geomorphological width function is exactly mapping to what is called a binomial multiplicative process. But in reality, what happens is this is not a river network, certainly not. But this has exactly represented a topology which is indistinguishable from the river network, for good or for bad. And this will have a bearing on what we say. So if I take these, this looks like a photograph, but it's not. It's a digital terrain map with proper doctoring of the elevations. You see the kind of detail, essentially ranging now with the LIDAR or LIDAR, if you want, maps of the next generation, for instance, to the order of 1 meter or less, to the order of thousands of kilometers. So rarely we have inspected in ground truth natural landforms over six orders of magnitude in scale, which is hard even for instance, for planetary phenomena. Now, rewind the thing and go into something which is completely different. And I'm talking about ecology now, an important contribution, a beautiful book, which I adored, hotly contested by a number of fellow colleague ecologists. It's called the Unified Neutral Theory of Biodiversity and Biogeography, which is essentially something which I'll be explaining you in a nutshell, probably know better than me at a certain point, but I'll do that with reference to an example. For instance, suppose you have like a lattice of this kind, one network shaped better, tree-like shaped. That is, every side has only one downstream steepest descent like descendant, okay? But it may have more ancestors, which you have here. So this is a network, or an OCN, if you want, optimal channel network of the, oops, I'm sorry, something that Ignacio and I, do you see this bar here, which is right in the middle? Okay, no, because okay, I put it here. Okay, I could. And in one case, you have a regular lattice, the so-called, this is the so-called von Neumann neighborhood. It's exactly is a square lattice in which a node is connected to its 4 nearest neighbors in coordinate directions. And I'm trying to run spatial ecology, sensu, say, Tillman and Careba. And let me take the exercise I'll be showing you, which can be done quite easily, is the following. It should be seen. Oop. Okay, oop, again, I can do. Sorry, I don't know what I did. Okay. What you see here, you start the dynamics of a system done in this moment. At each, a randomly select, suppose you have an initial distribution, say one node is one species, but is one color, okay? At any time, a randomly selected individual, that is, you pinpoint the place, a pixel dies, so you cancel it. And you replace it. Either replace it with a low probability called diversification rate, I'm sorry. You occupy this, this place by a new species, that is a color that wasn't existing before. With probability 1 minus nu, that means if it is like 10 to the minus 5 or something, that is every, say, 10,000, 100,000 trials you have in the system, the site is colonized with equal probability by an offspring of one of the nearest neighbors. Now, nearest neighbors in this case are really nearest neighbors, but is the four guys you have surrounding you in the phenomenal neighborhood. These two landscapes differ only in the definition of neighbors. One is a square lattice and one is a network, it's a tree. Now, you see what happened? That's the only difference between the two. What you see in here, you see patches are very different in shape and in color. And what is called gamma diversity is the number of species that survive at the end, the larger number being the number you have in there. Of course, there are technicalities on how you avoid boundary effects or how you take into account, well, nearest neighbor. But the fact is that that this result is quite interesting, because if you take, for instance, a measure, this is another picture of what you have, this is suggesting that in a sense, a network system, a tree system, is more shielded. You have an effect on the in fact, on the distribution of the resulting species. And why this is related to the neutral theory of biodiversity? Because you assume that, for instance, suppose you have surrounding you over 3 places, 3 red spots and 1 blue spot. Then you kill it, and with high probability, you become red. So if you didn't put any diversification or diversification ratio, whatever you want to call it, etc., that means that in the long run, you're going to have a single color, which is at time going to infinity, the species which was, or the color that was most abundant to begin with. But if you include this, which is a very important feature of biological evolution, the ball game changes considerably. This is neutral because you assume that you don't have stronger opinion. This is called the voter model in sociological studies, started from social sciences, this model. And it means that essentially the opinion you will retain will be the one of a majority of the nearest neighbors. There's no stronger opinion than others. It's simply abundance that tells you what happens in this case. In as much as there's no stronger colonizer, all at the per capita level, all species are equal, which is anathema to much of a niche hydrology, niche ecology, I think, which has been doing exactly the contrary for like 100 years since Elton's time. But what is the suggestion of a purely theoretical argument based on a neutral argument is that, look, guys, simply directional dispersal generates a completely different system. If you look at the abundance of a species ranked, you're going to have a much larger, OCN is the tree-like structure because it's not much in a network. It's called savanna, the other one, because of the kind of interactions that you can have in a substrate like a savanna in that case. And it means the biodiversity is completely different. And if you build this further, what you have is that you're doing the same exercises on one-dimensional lattices, 2D lattices, 3D, or networks, etc. And you measure a different feature. Here, in this case, is the number of time steps in which the same color persists in a place. That depends on the topology of the substrate very, very much. And this is a surprising result. And immediately what the ecological community will say, okay, that, of course, you're making the assumption this is neutral. Nothing is neutral in life. You have stronger predators, etc. And yet, this result resists a number of generalizations. For instance, it resists fairly well for the possibility of, for instance, relaxing the idea that you are sampling who's going to colonize you from the nearest neighbors. You can have a kernel for interaction. It could be a mean field. You could take the average, biggest abundance anywhere in the lattice. You can change the lattice as much as you want. And the result resists. Topology has an effect. And the more network-like the structure is, the more shielded it is, and the more the ecological thing results. So what we did, we said, okay, let's take it to the lab. I put up a wet lab owing to, God bless, EPFL that allowed me to do that. So essentially what we did, without much technology, okay, because you could have the robots that do that for you. So essentially you have the plates of this kind. And what we do, essentially we have protists, in fact, living in the thing. We have like, you inject an initial condition, which is known. And essentially you decide dispersal by having this done directionally. And what you have is these two guys, apparently by geography would be nearest neighbor. neighbors, but could be very far away because of the structure of the network. Like if you had to cross a divide to go into another valley. And no high technology. These are two colleagues now, used to be students at the time, and we did that on several replicas. And the result is surprisingly the same. There's nothing neutral in here, nothing at all. These are living communities with a certain number of species. And what you have is that in river networks, you have a higher diversity and you have a much shielded thing and much more uniform in a place which is a two-dimensional substrate of that kind. Or for the meta, and you can have, we had like a huge number of replicas, so you know even that this is not an artifact of a statistical thing by doing, I mean, serious things, etc. And if you start pulling in, for instance, like, hey, I say, look at the network, as soon as you go downstream into the river network, you see that the network changes its size a la Leopold, right? So you have like structures in which you find that there is a relationship between the size of the total contributing area, if you want, and the size of the channel. So what we did something in which we took the example in which we took the same amount, we took a Leopold-like relationships, allometric in fact, to generate the shape of a system. And in one case you put it hierarchical. In some cases you use the same volume and put it random or homogeneous, finding exactly the same result. So the message is, sorry guys, it's not the neutral assumption, it's something deeper. So the idea is that then why we started moving on species, that is whether large-scale water resources management plans can have something to say about the conservation of biodiversity for that matter. And you have systems which have nodes and a matrix nodes and connections. Nodes and connection means nodes are places in which you do have chemical, physical, biological reaction, or systems in which you have, in fact, ecological structures. Could be metapopulations, could be metacommunities and the likes. And I'll show you a few examples. And let me talk about my heroes, and that this is Ignacio Rodriguez Iturbe, the Pope of hydrology. This is Marino Gatto, he's a theoretical ecologist. And this is the late Ilka Hansky, whom I admire so much, who died early, in fact, of cancer. And he developed the metapopulation theory, which we've been working a lot on. So essentially, you take systems, you have nodes, which can be meta-communities, that is communities, or meta-populations, that is a separated population of a single population, of single species, functional groups, whatever you want to define. I'd be deliberately using a loose talk. If this was an ecological conference, I'd be way more careful, in fact. But the substance is exactly what I'm telling you. So this is a picture, for instance, that's interesting. What is LSR? Is local species richness in the Mississippi, Missouri river system. Iconic, one of the largest, one of the most well-studied, done by Bill Fagan, which is a fantastic ecologist working on fish in particular. And what you're seeing here is AARP, annual average runoff production, right? The stupidest thing which we do, the integral of a discharge passing through any particular cross section with a certain scale. And it doesn't take a deep statistician to see the more AARP you have, the more local species richness you have. So this is telling you something, but something about the driver, the hydrologic drivers and controls have a say, in fact, on a thing which is in this case local species richness. So what you do, you apply a game which is not unlike the one I showed you before, much to the dismay of many that looked for that. And this is, for instance, measured along the width function, that is the distance, upstream distance from the outlet of the Mississippi River. What a simple model of a type you have seen, neutral, that is, that treats every single fish as with the same per capita rate, no matter how shameful it is. And the exercise is as simple as the voter model. The only thing is that, of course, you take into account the kernel, in which essentially certain species may feel the downstream wind, the direction of the bias of the kernel, if you want to say. Long story short, data and the model tell you that this is a great predictor, which is essentially embedded in the shape of the Mississippi, Missouri. Or can we study biological invasions of fluvial substrates? It's called hydrochory, which is the propagation of species in aquatic systems. Well, interesting, this goes into meta-history, that is human range expansions, believe it or not. And I show you why in a second. Well, essentially what you have in systems which are basically one dimensional. That's the only equation I'll be showing you, in fact. But of course, my background is in applied math. So that's what and in a Galilean sense, I need to see something cast in mathematical form to pretend that I understood it. That's what, because it's a language that nature speaks, in fact. Now, in systems which are one-dimensional, that is in which width and depth and width are smaller than the longitudinal direction, river networks, no matter what. So 1D. So it's not a vector of the velocity of a system. It's a scalar, but it's dictated and determined by the boundary, something you can remotely acquire and objectively manipulate. So it's a 1D system in which X is the longitudinal coordinate along the river. So essentially a species diffuses, but depending on the thing, there may be a reproduction process, in fact, in there. So to cross the Mississippi from Chicago to the Chicago, there's a sense in that say Chicago doesn't belong. Untrue, it does belong. But what you have in the system here is that something which you have a logistic curve showing that essentially you have density, the density of population, the growth of population goes zero when the density is zero, and it reaches a plateau K of a carrying capacity over a certain thing. And in a sense, the time scale of a propagation, this is the speed of propagation, this is the diffusion coefficient, something like if a passive scalar moving through a river, moving into the system. And this is what knows is the giants of I mean, you name it, of founder of genomics, statisticians, R.A. Fisher and Kolmogorov, in fact, probably arguably the smartest mathematician of last century. They show that this equation here produces traveling wave solutions. That is, a wave which essentially propagates downstream the river, unmodified, in which essentially you see the square root. This is a velocity, length over time. This is a length square over time. So this is a dimensionally a velocity. Twice this this feature of the thing, which many of them are measurable, in fact, is the traveling wave, the celerity of propagation of an invasion front. Now, if you look at the what happens like in a stupidest thing, and I'm using piano for a reason, in fact, because it turns out that what characterizes, but what you see here is an interesting fact. When you have a system that branches downstream, you have lots of branching things, etc., you essentially waste time. You do not go as fast as you would go if you didn't have any ramification of the system. And the branching, in fact, doesn't feel what happens far away from the backbone of the propagation of the network, but feels the topology, that is the bifurcation that you have locally. So you have a system of this kind, and you can characterize this thing fairly well. So essentially, you see that a fractal, if you want to use it, a pompous term, but what readers do, in fact, whenever you have recursive bifurcation structures, essentially the bifurcations induce delays, which may mimic reactions and may leave like the time you waste in a second, and overall, they delay the propagation of a wave. Why this is important? I'll show you in a minute. So this is Kolmogorov fishes. Almost any tree, in fact, would do something different. It would be smaller than that. Theoretically, there's the Spaniards that work on that, Campos et al. Did a fantastic job. They calculated an exact solution for that. You get essentially what this is all about. Whereas the speed, as if the system didn't branch, would be different from that. And the example comes from a wonderful book. If you just now, in one of them, it's been just, it was a Princeton ecological monograph, now republished in the classics thing, etc. So it was very cheap. So I bought like 10 of them and I give them as a gift to all the coming students in the place. It's called The Neolithic Transition and the Genetics of Population in Europe by Ammerman and Cavalli-Sforza. And Luca Cavalli-Sforza has been one of the masters of genetics. So it's a model of a quantitative model of the colonization of the US, of a race to the West of the 19th century, and making the idea that the idea of a landscape heterogeneity and the need of water security forced settlers about fluvial courses, not in a radial directions, in any possible directions, because you need water for this, of course, for drinking, for energy, for transportation, and the likes. So the idea that the Spaniards has is to use the exact reaction diffusion model that Kolmogorov set in place in a system of this kind, and studying what happens in places in which essentially, if you look at the relative frequency of colonizing, which you have from the US archives, from the US National Library, in fact, it can give ingredients of how much time you waste in colonizing and settling. So essentially, you have a population growth model associated with the race to the west. And what is super interesting, in my view, is that the data are telling you that Fisher's prediction would tell you that no matter what, with the data you get from the archives, in fact, the diffusion and the archives, you would have like something 40 kilometers per year. The observed speed was 13. And almost any network you can conceive with any kind of tool, any kind of thing, etc., Peano's networks, OCNs, whatever you want, goes very close to that. Meaning that embedded in the branching structure, there is, in fact, the key for understanding how a process like biological invasions behave. And we took to my wet lab. I'm very proud of that, as you probably gather, because I'm a civil engineer by training, and it was all my friends were telling me, "Are you nuts? What the hell are you doing? It's a hell of a lot of work, and it's a wonderful thing." So we took and we generated these places to make protists run in a system, grow and run, to measure phenomena. And these are the pictures we took, by the way. I'm fascinated. It looks like Jackson Pollock or nearly so, but these are the wake colored artificially of every protist we measured in the system. To the point that we could actually now see from this small, the protistodrome that we have in the lab, back to the Mississippi, Missouri. And the zebra mussel invasion starting in 1990. Why Chicago? Because it was the veligers, that is the seeds of this major competitor, in fact, the zebra mussel, were brought in by the ballast water of some cargo coming from Ukraine, because in the European, it's a European, originally evolved in the European context, etc. That soon enough, he was then discharged into the Chicago Canal, and then into the tributaries of the Mississippi, and then propagated with density growing in time, as you see it here, throughout the entire Mississippi River basin. Like it or not, this is generates economic disasters, and quite interestingly, because that's important for the infections. At a certain point, you will see that in the beginning, this was just propagating downstream quietly, only to appear in funny place far away from that and starting the infection from that place. Why? Because recreational boats, not cleaned properly, were tugged up and taken up in completely different places, replaced in the water, again ballast water, to propagate the infection. Isn't what happens with waterborne disease at the same place? So what happens is that now the things become more complicated. I can give you details in here. So you have to make into account a model in which veligers, because you have sessile and free floating. So veligers flow are passive scalars that are diffused through the system. But when they reach a certain stage of maturity, they become sessile, and they become generated those clusters of zebra mussels. You can even have information on the commercial navigation in the Mississippi to get something in which essentially what you have is something like this, computed and measured are very much alike. So you can actually tell that in a small-scale Galilean thing, the mathematics are telling you exactly what happens. And you start introducing what you call the multiplex network. So you have a basic network in which things propagates, but you do have shortcuts generated by something, in this case, commercial boating, and the transport of seeds of the infection, quote unquote, in completely different places, for which you may have some proxies. So that's, I'm rapidly coming to my, well, not rapidly, I still have 10 minutes or so, but it will buzz when time comes, Jeff, don't worry, and I will wrap up. So the idea is that now you have systems in which you do have a topology, you have a nodes, and you have the connections. And in the nodes, you can have individuals, you can have metapopulations, you can have metacommunities, you can have disease demography. So essentially, I'll be showing you now, we soon realized, in fact, it was, it's not a joke. Ignacio and I took several walks around the pond of a Princeton Institute for Advanced Studies, because it was a place in which we like to go, etc. And all of a sudden, in an afternoon, in fact, maybe 15 years ago or so, we saw that everything could be done, species, populations, and pathogens, because they are mirroring exactly the same phenomenon, which is you have transport of something, mass of diversity of features, etc. You can have delays due to the structure of the network, and you can have reactions in the nodes. So I'm now to the disease part, getting back to what is important for water security. The example I was making about the clear relationship that meta-analysis put forth for the correlation between the expansion of irrigation schemes and the expansion of the burden of disease of this disease, which is largely 90% of that is Schistosoma mansoni and hematobium are distinguished. The two flatworms, the parasites, are distinguished depending on whether they colonize in either in the bladder or the intestine, and but essentially the dynamics very much the same. Which end the thing, etc. And as I said, this is a neglected tropical disease because nobody dies in there. But it's a poverty reinforcing disease because the learning impairment is very significant because of the debilitation of that, and it doesn't give you acquired immunity. So how could you put a price tag on the burden of disease compared to the effects of agriculture? You're dead unless you can predict how many cases you're going to have. That's what we will try to do here. It's a complex ecological cycle because this is my hand. In fact, I took a picture. This is the kind of the freshwater snail that works as an intermediate host in the system, and I show you in a second how in fact the migration follows in this case. And this is the kind of, by the way, the signature of a disease is these particularly these bellies of the young guys that are kind of inflated and as I said, and what happens is that what is interesting about this is in fact, for instance, the guys or students who did his field work, whose results I'll be showing you in a minute, got it actually. You know why? Because once and only once he removed the gloves to pick up the scissors that he had dropped in the water. Just this movement, and he caught it. Of course, praziquantel, and he was wiped out, but not in rural Burkina Faso. Oops, I'm sorry, it's already 3 minutes. I really chatted too much. So this is essentially, it's a complex cycle, and what you see in here, that essentially you excrete eggs hatch, they become miracidium. Within a few hours, they have to infect the proper snails. And then later on, these guys are a pump of millions of the cercaria of flatworms that can penetrate the skin fairly easily. And so what I'm saying is that, so burden disease can be calculated. What I'm showing is that quite fast, in fact, the disease curbing role of a local abundance, you can have like measures that can complement water resources, in fact, management schemes, natural predators, like certain shrimps, for instance, could do the job for you, certain poisons, or some controls. You can have water running. And of course, you could prevent the kids from flowing into the water, very complicated, right? About all of the distance of that. This is the kind of data, well, the kind of data that we have. And I've you see, these are the kind of environments which is sampled the thing. And only the same, but before I go to the conclusion of the traveler party, fieldwork in Africa, you know, that's what we did in our place. This is a receipt that my guys took at Il Cappuccino in Ouagadougou in the morning of this, what is 16 January of 2018. At noontime, terrorists broke into the Cappuccino and killed 35 people. Seriously. So I was scared to hell, so I wrapped up and we said the trouble of parting is exactly what we have. I'm sorry, no, I wanted to show these before I quit. Well, long story short, with a few tools like eigenvalues, we can show what happened if, for instance, if you took out like 30% of the small dams, and there's a probability of disease, you see that the white spot increases significantly. So there is a hope of getting actually a thing. And I won't speak about cholera because I would have spoken. Well, let me do the last two then. Only to say that this is cholera in Haiti, and how in fact we can actually data and model cholera. Epidemic cholera is something on which you do the same thing. I'm simply showing you, look at the data simply, not at the simulation, which is called involved and it takes some time. What you see here is what? You see the river network, whether you like it or not. Of course, punctuated by issues which are of course mediated by human mobility, or something which you can talk about, or things in which technology can help us significantly, like for instance, whether we like it or not, the ownership of a cell phone, which is a localizer, is way less socially biased than we might think. I've seen with my eyes in Carrefour, the most dilapidated market of the poorest country in the world at the height of the epidemic cholera. I saw a young lady, and I was into a fortified car because they could kill you to steal your camera or to steal your watch. I saw a lady showing a Nokia 1900 and getting cabbage in return. That makes you worried about water security. Okay, you can't secure the water, but you can guarantee almost anyone in Haiti, the poorest country of the world, a cell phone. There's something funny there. So here are my conclusions, and that yes, there are eco-hydrological footprints of rivers, ecological corridors. You can see that in the distribution of biodiversity. They make robust, their sheltering effect has an effect on the conservation of biodiversity, so they could be guiding next generation or water management plans 3.0 or something. They can, from peaks of prevalence in waterborne or water-based disease, I haven't really given you details, to large-scale patterns of species abundances of biodiversity, or susceptibility to biological innovation. As Mary Parlange once said, it's all in the water, actually, and in the particular geometry of a substrate we allow for ecological interaction. So my search and my interest scientifically and hydrologically is towards a fair distribution of water. I think it's a moral imperative, in fact. That is, quantitative evaluation of ecosystem services are necessary to rethink social equality, and in particular, in my view, global water security. Thank you. I'm 1 minute late.
Thank you very much, Andrea.
1 minute late, I'm sorry.
No, you're good. This is the point where we open it up for questions, either through the chat or people can just unmute themselves. And while we await comments and questions, it was interesting seeing zebra mussels. I used to own a cottage in the St. Lawrence River when I taught in upstate New York. And for me, zebra mussels were a godsend because I like to scuba dive, and they made the St. Lawrence River crystal clear.
Exactly,
and,
they filter everything.
All the shipwrecks that I love to go and dive on were now beautifully visible with you know, clarity like the Caribbean. It was incredible. Here's questions come into the chat. What are the next challenges under this approach?
Well, okay, that's a good question, and I can tell you that serendipity in science is quite important. Mind you that, believe it or not, our group, published a paper on COVID-19, which is one of my highest cited paper, or nearly the highest cited paper, etc. It has something like 600 citations in a year because it was published in March 2020. And well, of course, that reflects the size of the crowd of the people working on COVID, etc. But what happens is that what we have learned in this case was immediately, in as much as you had those high fashion industries converting lines of production to generate masks during the COVID. So we soon realized that the spatially explicit tools we used, including human mobility, including all the things that you need to have to characterize epidemic cholera or endemic schistosomiasis, are essentially the same math tool. So the idea is that what we have learned here can be expanded, can be generalized, and can be, without exaggerating, could be even be useful, right? And as it was famously said, that models are fun and sometimes even instructive. So in terms of the next challenge would be to provide enough know-how to allow, for instance, policymakers, for instance, to say, okay, now I'm planning water resources in a large river basin. May I please allow conservation of biodiversity in there? Because it is important. We're losing biodiversity rate, which is amazing. And of course, not all ecosystems are water-based or water-controlled. Many are nutrient-controlled, many of the things that, but quite a few are. And river, much of the biodiversity of the world, think of the Amazon, for instance, is stored, in fact, in river networks. So the first thing is just tackle one at a time. It could be, for instance, infections be seen in the shape of a river network? Then you will know where to make provisions to make defense, etc. Can they use to be, the type of approaches we have now that we know when the constraining factor of a shape of a ecological substrate for interaction is as well known as you have for river networks, you can probably say something about, again, biodiversity protection, about river management with an eye on what's going to happen next, to the pricing the planet in a sense. A quantitative evaluation of ecosystem services requires knowing what's going to happen. So I think next would be marrying eco-hydrology and environmental economics, I think. Did I answer your question?
Yeah, I think that's good. Other questions? People can just unmute themselves or type into the chat. I'm reminded of, just while we wait, the work of Howard Odom, and one of his students, maybe his last student, Charlie Hall, who was a colleague of mine, again, when I was in New York. And Charlie was denied tenure the same week he had a cover article of in Science magazine at Cornell. So this will be topic for maybe our next half hour after the top of the hour when we talk about early career issues. But anyway, that was a big theme of their work, right? Challenging neoclassical economics, trying to bring ecological thinking into play.
But for instance, Jeff, you see, Hubble's work on the neutral theory is despised by, I say, good 45%, 75% of the ecological community. Why? Because we're a niche ecologist, but he's studying the specificity of certain species, right? Well, this guy is telling you, look, neutral process doesn't, neutral pattern doesn't require neutral process. So you can assume the blasphemy that all species are at per capita level are equally performing, and still what you get through the system, and it allows you to get nice results of statistical mechanics done by proper statistical mechanisms, of course, etc. But yes, path breakers, Elton in Oxford did so. Ilkka Hanski did so in Finland. Hubble did that while he was at Princeton, and then he's now peregrine through the system here. So thinking out of the box is very rewarding, but in a limited number of cases. Yeah. So it's safer for the rest of the discussions. That's probably safer to have like two ovens, so as to say.
Any other questions before we transfer to the early career group? I'm looking for hands up or comments in the chat. All right, Andrea, I think we've got 2 minutes to the top of the hour, and this is perfect timing to thank you really for an exciting potpourri of topics relating to ecological networks. And I think this will propel us into this next section. We might be on for 15 minutes, a half hour. We'll see how the discussion goes with the early career folks that are interested in staying on. So I'll bring the formal part of this seminar to a close. Thank everyone for joining us, participating, and many I know are participating remotely. And thanks, Andrea, again, for a really exciting talk.